Hey everyone, it's Mr. Wolfe here, and in this video I'm going to be going over the first part of protein synthesis, which is called transcription. So this process is going to start with DNA. And before we get started, let's kind of just get acquainted with our setting here. So this circle area, that's going to be our nucleus. And one thing to notice about our nucleus, and we actually have not talked about this before, is that there's these little holes here. We'll explain why those are important in a sec. So we have our nucleus, which means outside the nucleus, this area in general is just called the cytoplasm. And then if we go a little bit lower, even though this guy's not involved in transcription, it's going to be involved in the second process, we can label it now. This is going to be our ribosome. So back with protein synthesis, remember our overview, we're going to go from DNA and we're going to make a protein. That's going to be our ultimate step. However, before that, we have to do something first, and that is going to be the process called transcription. So transcription, what we want to do is we want to take DNA and we want to make an mRNA molecule. So how does that go down? Well, in our nucleus, I have a DNA molecule that I drew, and you can kind of see that I highlighted or zoomed in to a specific sequence. Maybe this could be the gene that makes lactase. That's the protein that digests the milk protein when you drink milk. So here our sequence is right here. And what's going to happen is an enzyme is going to come in, and it's going to open up our DNA molecule like this. So this is caused by an enzyme. And now that it's open, we have just a bunch of these free RNA nucleotides floating around our nucleus. And they can come in, and the same enzyme that opened it up is going to grab these nucleotides and place them to their corresponding bases. So for instance, once we open up the DNA, we have a T here, so that enzyme is going to grab a RNA nucleotide that's an A and match it up with a T. Now, remember, there's no T for RNA. Instead, the nitrogenous base is uracil, so a U goes with an A G goes with C, A with T, U with A, C with G, U with A, C with G, G with C, U with A, and then A. All right, so you can start to see that we're making our mRNA molecule. Now, what eventually happens is this mRNA molecule detaches from our DNA molecule, and it's on its own. So I'm just going to redraw our RNA molecule over here. A, U, G, A, U, C, U, C, G, U, A, A. And let's just label this. This is mRNA. Once this happens, once the mRNA molecule leaves, the DNA just winds right back up. Okay. So now that I have my mRNA molecule, I have this message, my master plan, my DNA could stay in the nucleus, but this message of just one gene, in this case, let's say we're making lactase, that enzyme that digests the milk protein, this can now leave, I'll draw this in red, and go out into my cytoplasm. So that's going to be transcription in a nutshell.